Hello, good evening. Thank you very much for the warm welcome. I'm Jan. I've actually just submitted my PhD thesis in September. I'm still working with Professor Paul Dupree in the Department of Biochemistry. And I wanted to tell you a little bit about my work on wood structure and possible application of some of the discoveries we did on degradation of this material. So we've got some pictures of trees, of pine trees here. But I wanted to start by drawing your attention to this curve, which may seem familiar to some of you. This is the amount of, well, the concentration of carbon dioxide in the atmosphere. And most of this increase, which we are facing now, is supposed to be, or well, is believed to be man-made. However, the interesting aspect that I want to point you to is this annual variation in the amount of carbon dioxide that we have. And this annual variation is one of the very first observations that we've made on the amount of carbon dioxide in the atmosphere. And actually, it is all because of the activity of plants. So in the summer, or in the spring, actually, when plants start vegetation in the northern hemisphere after winter, we get a decrease in the amount of CO2. So it's showing you how much difference plants can make. Actually, majority of this CO2 is going into wood. It's locked in wood, in timber, as polysaccharides. And it is estimated that if we were to increase our forest reserves, our forests, by 20%, we would match what we are releasing into the atmosphere, which is possibly a manageable goal because we've increased European forests by 6% since 1990. So this is wood. We actually, in the lab, are working on a model plant, Arabidopsis, which maybe doesn't seem similar to these hardwood logs, but at the microscopy level and also molecular level, it's quite similar. So you see here a hardwood cross-section, and the defining feature of wood are these thick structures around the hollow vessels, and these are cell walls. These are plant cell walls which are made predominantly from polysaccharides, so long chains of sugars connected one to another. And I wanted to tell you a little bit more about these structures and about how we've modified them. So in order to visualize that, I brought tonight with me a model, a molecular 3D printed model of two different structures. One of them is cellulose, and it's what you see there in the orange. And another one is xylan. That's another polysaccharide that we have in cell wall, and we know it binds to cellulose. And what we found out is that this xylan chain is made by a range of enzymes. Enzymes are machines that are present in plants that make something. And we found that this one group, GAX enzymes, they're very important. So in our work, or in my work, I've been looking at the resistance of this material to degradation. I'll pass this around so that you can familiarize yourself. I'll be talking mostly about these sticking things out from the green stuff. This is glucuronic acid on xylan. Glucuronic acid is quite a mouthful. So we've been looking at the removal of this glucuronic acid from plant biomass using genetic engineering. So we've been changing plants by removing certain enzymes which make this biomass. And we've noticed that if we remove glucuronic acid in this particular mutant, we increase the amount of sugars that we get. We increase the glucose release, one of the sugars, by 30%, and strikingly another sugar by as much as 700%, so a massive increase. And this increase, we've been able to coin it into a useful product. So we've used an interesting system in which we can turn these sugars into a biofuel. So I don't know if you're familiar, but one of the problems in biofuel production is that not all of the sugars can easily be used. But we figured a way of using both sugars, glucose and xylose, in a bacterial system and fermenting them into ethanol. And actually, if we use our transgenic plant material, we can get as much as double the amount of ethanol, so possibly a good feedstock for production of bioethanol. And interestingly, or importantly, these plants grow the same as any regular material, so there is no yield penalty, no decrease in the amount of biomass that you get. And because of that, I've been interested in softwoods. So I was fortunate to identify two enzymes in softwoods that catalyze addition of this branching that determines the resistance of wood to degradation. And now we are trying to look into actually modifying the softwood material to have it growing the same, but being more easily processable into fuel or paper or other advanced products. And these are pretty much the conclusions. We've developed a way of producing highly digestible material, which can enhance production of fuels, of paper, or also of paper. It is a transgenic material, and that's worth noticing and taking into account, and I'll leave it up to you to decide what do you think about it. But it can be a very good way of dealing with some of the problems we have with climate change, for example. And these improvements can contribute to a range of industries. And we think in not only traditional things like paper, but also novel industries like, for example, nanocellulose, which is getting bigger in, for example, food industry. So I just wanted to give you some perspective on the grant I've been given. So I've been fortunate, and I'm really honored to be awarded the Student Award for PhD research. And the money I've been given, I used to visit a Joint Bioenergy Institute, which is a research institute in the US looking at degradation of plant biomass, a really prime research institute. Also, while there, I've attended a conference, and actually, the poster which presented this work was, wouldn't be possible without the support from the society, was awarded by the peers Best Poster Prize. So, very, very fortunate. Thank you. So, all of this work wouldn't be possible without the help of my friends from the Dupree Lab in biochemistry, and there were very many different collaborators around the world. And of course, the society supported my attendance here, and some of the work I'm doing now with the friends from California. So thank you very much, and I'm open to questions.
Right, time for a couple of questions, if they're really good questions. Gentleman down at the front here.
That first slide. Sorry. Thanks. The first slide was the CO2 going up like this and the trees doing this, the plants. You said 20% increase in trees might cure this. Well, the amplitude of that was tiny compared to this, so how do you reconcile that?
So the only reason for the amplitude is the difference between the amount of vegetation between the northern and the southern hemisphere. In the summer, or in the spring here, it's winter down under. And if the amount of vegetation would be exactly the same, these effects should cancel each other out. So the variation that you see is only the delta, the difference between the amount that it's locked between the hemispheres.
And what's the real amplitude?
The real amplitude, I don't know the actual numbers, but the wood that's locking this CO2 is the basically biggest repository of carbon dioxide. And this increase, well, I'm not proposing a 20% increase in forest coverage because for agricultural reasons it would be impossible. But it can be part of the solution. That's what I'm suggesting.
One last question. You're all nervous now about whether you've got a good enough question, aren't you? Well, we'll let you off, and you can be a little braver when we get to the main